Hi everyone. Thank you for having me over and listening in. Thank you, Hans and Peter, for inviting me to speak today. So glad to meet everyone virtually. I'm Yilin Chen, a third year PhD student at Stanford with postdoc Huang's lab. So I mainly work with computational protein designs and protein representation learning, as you'll see. Um, yeah, that's basically about me. So I'll just go ahead and talk about the background of the paper. So today I'm just going to talk about our new work SLAE. That's shorthand for strictly local all-atom environment for protein representation. Okay. Okay, so the overview of the talk will be just having these sections. I'll give a little bit of background on protein representation. Hopefully everyone is somewhat familiar with proteins at least. I'll give an overview of the model and then talk about downstream tasks and then latent space analysis of the SLAE model. And finally, the conclusions and some future work. Okay, slide. So a quick introduction of protein representation learning. Why do we do it and how do we do it? So two pipelines that people who work with computational protein designs are interested in are, well, first, of course, the protein design and also protein property predictions. And these are some oftentimes related. So in computational protein design, you will have some model that generates you a backbone, design some sequence, and most importantly, I think the step that's interesting is having a computational filtering or oracle step that allow you to filter out your designs and tell you before you go into the expensive experiments if your design is good. Okay. And then we'll also have the protein property prediction problem where you give a protein to a model and that it predicts some properties you're interested in. For example, the oracle that we want in our designs. So there are also other things that people do with proteins and machine learning models. We can talk more after going over the model itself. But I forgot to say, but feel free to ask any questions during or after. So feel free to interrupt. I just want to keep this casual. So the next question is how do we featureize a protein to allow those computational pipelines, right? One of the most popular or hot topic right now is language model. So protein have a protein language model like ESM2, PROGPT2, and there are a lot of other models that basically follow some sort of language model objectives. Some are masked, some are all regressive, and to learn a representation. And what this learns from seeing so many sequence and with a transformer that learns pairwise relationship is basically evolutionary statistics. And people can use it to predict structures, for example, as in the ES4 case, but also other properties, right? The other modality that we have are structures, right? So currently, I think a lot of popular models are backbone graphs. The graph is a natural way to represent the molecular structure. However, the protein contains thousands of atoms, so people usually just abstract it to graphs and maybe with some featureizations of the side chains, such as torsion angles. And then we'll use like a GNN encoder through some pre-training and auxiliary task to feed the model information about how to understand the protein, and then use that model for downstream tasks. So what these type of model usually captures are backbone geometrics. However, both I think makes an abstraction choice, which I think is worth considering. So they intentionally discard explicit atomic details. Okay. Okay, next slide. But I think atomic structures matter. So I came from a biophysics background, so this is very natural to me that when I look at protein, I think about all these side chain atoms interacting, right? In the middle boxes, the atoms are interacting with each other. And this is actually, I think, what really determines the function are these sidechain details that we abstract away in those representation model I just talked about. So these are sidechain packing, the hydrogen bonding, electrostatics and solvation. This affects a lot of aspects of proteins like stability, binding, enzyme activity, so on and so forth. But like I said, reasoning over and representing so many atoms in one protein is very expensive, right? So the question is now, how do we retain atomic level physical information while trying to keep computation and representation really manageable with these large scale models? Okay, next slide. So I would like to step away from proteins a little bit and talk about representation learnings and computer visions. So the question here is, how do you model understand image? So I think all the image have some sort of semantics, right? What this image means, what is it about? So for our audience who are more familiar with computer vision, that you might know CLIP and Dino, which are the two most popular models. So on a high level, the CLIP model try to match an image and the text. So through contrastive learning, it learns what this image is about. Well, I like to talk more about Dino. Right? So Dino is a self-supervised model, meaning that it does not have labels, like this is a cat. So what it does is it tries to learn a representation of the entire figure from these patches. Okay. Um, Stino basically showed us if you force a network to recognize a zoomed in patch, say an eye of a cat, and know that it belongs to the same cat, the model learns the semantic of what is a cat. So whether or not, so without being told that this is an actual image of the cat. So this is trying to get information into the tokens of this image. So the takeaway is that you don't need explicit labels if you design the right learning signal. And local context can define global learning. So when I was developing SLAE, this sometimes come into my mind. I feel like this aligns very well with the philosophy I had. Okay. So I guess now bring us back to proteins. So I want to build something toward a compact all-atom protein representation. And I think, or this is my hypothesis, the semantics of a protein are encoding each residue's local chemical environment. Right? So how do we learn the semantics of protein? So we just apply the similar logic from Dino to atomic system in this case, right? We hypothesize that what matters is what's in these little figures, what are around each atom. So essentially this breaks down into pairwise atomic interaction. So it's sort of like a first principle way. The type of atoms are interacting with each other and their distance and orientation. Right. Okay. I'd like to step away again and talk about autoencoders. So I think people who think about autoencoders think about the figure on the right. Like you have an input, you feed it through an encoder, squeeze it down through an information bottleneck, and have the decoder to try to reconstruct what you gave it. Okay. But I really like what's the explanation, sorry, the explanation on the left is that the autoencoder is trying to learn a function and a submanifold of the data, which is the latent space. So it's a learner of the manifold. The encoder maps very high dimensional data to low dimensional latent X. So in our case, the high dimensional data would be thousands of atoms in this point cloud, and their coordinates, right, in a protein structure. And our latent is something that we're interested in that's easier for a model to see. Okay. So we always, through this, learn a k-dimensional submanifold of the data space, and that serves as an information bottleneck. But the question is then, like, what information should this submanifold capture? That's an expressive representation of protein. And how do we enforce it? I think the answer I think I explained are chemically meaningful local environment, but they have to be physically consistent. So according to the first principle, and you should be able to reconstruct the global structure from it according to the design of autoencoders. Okay. So this comes to my architecture of SLAE. This is a overview of what's going on here, and there's a lot. And this is the classic encoder-decoder representation, right? Um, I think what I want to emphasize here is that we're giving this autoencoder a very hard task. So it has two parts and it's decoupled. So it has a local encoder, which only sees what's within the local neighborhood of each atom. And the encoder needs to reason within these neighborhood only on the information that's chemically and physically reasonable. So atom type, distance, and orientation. And then this gets boiled down or minpoled into each residue. So now the residue token is a combination of what the atomic environments are. And then this is our information bottleneck. And then after that, we have a global decode. So the decoder actually never sees, sorry, the residue latent never sees the global information. But the global decoder is tasked with this very hard problem of building back those bubbles that we deliberately broke apart in the encoding process. So the decoder sees like a series of convolved tokens of what's only the information of what's within these bubbles, and it needs to piece them all back together into this very complicated protein structure. Okay. So I'm going to dive a bit into the designs of the architecture. So the encoder is designed as an IC3 equivariant local environment graph neural network. So same figure. And we're building these graphs only within 8 angstrom and with directed edges. So this is very similar conceptually to some of the machine learning force field method that learns interatomic potentials, but it was never trained on DFT. The goal is to train for downstream reconstruction. So the model have these internal representations of the scalar, which are atomic species and interatomic distance of an atom pair ij, which you just do an MLP2 body of, and then with like a projected distance. And then they have the equivariant part, which is a weighted projection of each unit ij vector, so the direction of that vector. And then at each layer, you basically update the equivariant state of the center neighbor pair ij with all other atom neighbors, so IKs, in its local radius cutoff. So this is
And
sorry, yeah.
yeah, just to understand a few sort of details here. So is this, are you doing this for every single atom that exists in the protein or every residue that exists in the protein?
Every atom. So this
Every
is on
atom.
the scale of thousands, but you can do this parallelly because
Sure.
the edges are directed, right?
Sure. Um, and then what kind of sort of things do you know about the atom? Obviously, like the atom type and distances, but like, do you know like this belongs to this particular like residue? There are the covalent bonds, whatever.
No, there are no other information, so it's only atom type and distance or
Okay.
and direction. So I want to boil it down to as simple as possible, like first principle way. So I do not give it residue type, residue number, covalent bond, or anything else.
Okay. But the distance presumably tells you a lot about the kind
Right.
of bond. Yeah.
Yeah. So the model will learn that very fast. Yes. But so I think like those information we should allow the decoder to figure out. In that way, you can squeeze that information back into the latent so that it is meaningful and expressive. So basically, at each layer, you update this atom pair with this local neighbors information. So what essentially in these edges are information about all the neighbors that this atom have. And then you can aggregate all these edges into the atom itself, and then you minpol all the atoms in one residue to get a residue latent. And the details of this, a very similar implementation can be found in the Allegro paper of this is the MLFF model published in 2023. Okay. And then so once this is done, it gets you can extract like per atom embeddings, which are 128 dimension. You can also get per residue, which is also 128, and you can just min pull into one vector for the entire structure. I'll show later that all of these are helpful in some ways. And then the residue latent, now per residue, 128, are feeding to a global decoder. So these are essentially tokens of residue chemical environment. Okay. And then the global decoder is just a very simple transformer. So the transformer allow it to reason over long range interaction that the encoder doesn't have. So its goal is to retrieve that and exchange information across the entire protein and enable the reconstruct of coherent backbone and sidechain information. So one of the things that we find really, really helpful was to have auxiliary task. Precisely the pairwise energy terms that we also regress. So this shared transformer backbone have three heads, one for sequence and one for coordinates, and the last one is for pairwise energy terms. So these are Rosetta scores that we pre-compute on all the training structures. And that accounts for solvation, hydrogen bond, and electrostatics between all pairs of residues. So that allows information to be kind of learned in this latent to make it physically meaningful. Okay.
Why do you think that is? Like, because like presumably you have the positions, sort of, at least the relative positions, of all the atoms, which can give you a sense of the residue position, and then you're doing a protein folding or like, well, not folding, just reconstruction.
Yeah.
Like, assume it's the folding, which is like you just have residues and you want to fold and put the final structure. Like you have all the information available in a sense, and you're just trying to like, get that out.
Yeah, so I like the folding analogy, but this is not trained on any sort of MSA or sequence. So it does not have any coevolutionary information, which makes like the folding part hard. So it needs some sort of learning wheels. For example, it needs to learn what hydrogen bond, for example, in the backbone to be able to reconstruct those secondary structures.
Yeah,
And
but I mean, I would think folding is even harder, right? Because like folding, you just have a sequence versus here you start with
that's
core
also
like
true. Yeah.
positions. Yeah.
Yeah. I think it's a combination of the philosophy and also it was originally designed to help smooth out this latent, which will show some experiments later on. But it was originally introduced as a smoothing kind of term. So so essentially what you're predicting is this heat map on the right. So if you were to if the decoder can get this correct, it would know that say, oh, these two have a strong hydrogen bond, so they should be close to each other. And that's how you sort of get the per atom thing out from these mean pool residue tokens, if that makes sense.
Hmm.
So I like to think of the residue as a very convolved um, representation of the atoms. And to get things out, it is kind of hard. I think my um, are you still seeing my slides okay?
No, I think it's been disconnected.
Yeah, I think my sorry. The connection just dropped. I'm sorry.
No, it's all good.
So I hope that okay, can you see that again?
Yeah. And just to understand again, so this is you're doing from the residue level embedding, you're getting atom level structure.
Yeah.
And do you like well, when if you like evaluate just alpha carbon sort of structure, do you also need additional terms to get to like for that to be good? Or like I'm just trying to understand, is it the global structure that it is having hard time getting, or is it the local structure that is having hard time predicting?
Um yeah, that goes back to the training. It's a global structure. That was really hard in
Okay.
training. Yeah. So precisely those like long-range interactions. So essentially you
Uh-huh.
have these bubbles that the decoder needs to know which bubbles are connected. And it's like
Yeah.
a huge puzzle you need to figure out. Yeah, so the essentially this is trained as the combination of these three loss terms. Um, because we also don't know the sequence. You first need to decode out the sequence. That serves as your Atom 37 mask. So you
Hmm.
predict all possible atom coordinates and you use your predicted sequence mask to get your final coordinates. And then you also supervise with this energy loss.
So I have a question.
Yeah.
Exactly to your point, you mentioned the long-range interactions. I'm wondering how can you capture that if your representation is so local and at atom level? How does the model deal with that?
Yeah. So the residue is a combination of all its atoms, local 8 angstrom. So it is actually a reasonable range to start with. But I
Mm.
also would say that a protein is just a combination of these local interactions. So if you
But
know
what
like
do
these two atoms are close, and then you're just trying to piece out the global structure back from these contact map, if that makes sense.
mm, but what if you had like a really global or like really folded up protein where different like tertiary structure motifs might be sort of like close to each other? That's like very far in the sequence level, but structurally in space it might
Yeah.
end up interacting.
Yeah. So that's like essentially what the decoder is trying to deal with. And it
Yeah.
now sees all these local bubbles. And maybe they have some shared feature suggesting that they're interacting, or does the energy objective that the model learned that these two things are probably close together,
Mm,
even though they're far away in the
mm,
sequence space.
mm.
Yeah, I hope
Yeah,
that.
so the question from Tim, which is does the encoder know the residue atom indices of the nearby atoms, or just like what element they are?
There's no position encoding no indices.
Okay.
So it's purely on atom types and distance.
But presumably, like that's okay to include, right? Like that
Yeah,
doesn't
that is also
hurt.
okay too.
Yeah, yeah.
Yeah, I was just thinking that it wouldn't help that much.
Okay.
And the atom numbering is kind of random. Yeah. So yeah, that's the design of the OM encoder. Any other questions? Okay.
Hi, Yilin.
Um,
Oh, my slide.
Oh,
Sorry.
yeah, I had a question on
Yeah.
hi Yilin.
Oh,
I have a
hi.
question on training, and maybe I missed it, but did you mention the data set? And is this trained on PDB structures or like the
Yeah.
whole sort of AlphaFold universe?
So I okay, let's talk about it here. So I have it in my speaker notes. This is trained on a set of synthetic cath structures, meaning that we took the cath fold, designed a sequence, and then fold it back. So that way we enrich the cath. So that gives about 30,000 structures. The reason for that was that was a cleaner data set than just training on a PDB. So the PDB has a lot of artifacts and needs careful filtering. So for an autoencoder, we decided to just use a synthetic data set. However, this is also evaluated, so trained on synthetic cath, evaluated on PDB. So trained so the performance on like actual structure, PDB structures are about in the same ballpark. So it's not that the model learned some sort of bias from those synthetic structures. But yeah, good question.
Thank you. And performances as measured in, say, the RMSD of the reconstructed
Right.
structure. Okay.
So these are measured in all atom RMSD. So since we're talking about training, we did some experiments. So the point that was raised before, the energy term, was actually very, very important to the reconstruction. So just removing the energy term, the model does not train. So it could not learn how to get the all and the structure back. So it's kind of a training wheel that we could not take off. And then we also had different tests of the radius, like the other question that was raised. So we tried 4, 6, 8, 10 angstrom. So anything larger than that, the compute kind of blows up. But for the smaller radius, we reason that it does not have enough context for the global structure to be built back. So I think the 8 angstrom we found is kind of the sweet spot here that gives us the
And
best reconstruction.
and for reference, like what's the RMSD for like just protein structure prediction in
Protein structure
general?
as in AlphaFold.
Like any all atom sort of model.
Yeah, that's sort of in the ballpark of 1 to 2 because that's also the resolution limit, right? Your training structures sometimes have a lower resolution than that.
Hmm.
So I think this is well within the range of accepted errors.
Sorry, Marianne, on the energy term, how do you get that for training? Is it it's based on the structure and you compute the pairwise for like even including the global interactions or the sort of long range interactions?
Yeah,
Using is it
yeah. So
for the neural network
so
function?
n by n essentially. So
And
we
then,
have
okay.
this heat map that's pre-computed for each of the training instances.
And it's each interaction or just the ones that have an H bound?
All the
But just
interactions.
okay.
So all pairs. So sometimes if they are not interacting, they'll have 0.
Okay, got it.
So the
Okay.
model also learns, oh, those two are not together, or those two are not near each other in a helix.
So like no long range Van der Waals type interactions, but the local, I mean the global matrix of local pairwise H bonds.
Yeah, that's right.
Thanks.
That's right.
And then some questions from the chat. There's one, how you do minimization? Sure what that means if you want to clarify a bit more on the question, Harshita. And then Chris says, do the structures in the training data represent multi-merge structures? And do you capture the information across individual chains of complexes, for example?
Yeah, that's a really good question. So we trained on monomers for now. So there's plan on training for multiple chains. However, I would say that the encoder does not see chain IDs. So the information you gain from the encoder actually works if for multi-chain interfaces. I will show an example actually later on. But yeah, so the reconstruction right now is not that good for multi-chain because we're not trained on that. Meaning because there's a chain break if you have two chains, and that's not well handled, as that's kind of
Um,
because of the decoder, right?
can you explain that again? Why it wouldn't be?
So the decoder has the positional encoding, right? For that's undefined for multi-chain in our current setting.
I see. So that's the position encoding that you add at the
Right.
residue level for the
Right.
sequence to the transformer, not because of like not because of the encoder.
Yeah, so for the
Yeah,
decoder.
yeah.
So we did not train with multi-chain in mind at the time. So it would not be able to figure out the chain break, for example. Yeah, I hope that makes sense. Was there
Sure.
another question?
Yeah, I can
Yeah,
you hear me?
yeah.
I wanted to ask you, do you do the energy minimization after when you have the all atom structure and
So
then
was it
compute
for
the
training?
RMSD? No, like after when you have the all atom structure and I see the results on the slide. So do you
Yeah,
do energy minimization and then compute the RMSD, or
no.
is it just the pure structure that?
It's just the output. So
Okay.
this is just the sequence maxed Atom 37 coordinates. So the
And
hat actually just directly predicts n times 37 by 3. And
okay.
that is
Okay.
used to compute the RMSD.
Gotcha. And do you you mentioned there are three heads of the transformer.
Right.
So do you weigh them differently or they are equally weighed?
So it's weighted in training. So we start training by upweighting the energy term, actually. We find it really helpful in early stages. So once the structure gets good, we kind of just downweight the energy and let it focus more on getting the side chains right. But the three heads are parallel. But their learning is gated by this weighted loss term.
And then additional question from Marco, more about disordered regions. So do you have uh do you study, I guess, the ways in which local short disorder regions are affected? Or do you see anything there? Um,
Yeah, so we didn't do a specific analysis on disordered regions. However, they normally, just say that, they normally decode fine, as the latent have some sort of knowledge of the phi psi angles, for example, and is able to get a basic understanding of which two residues are close to each other, not necessarily just memorizing where the secondary structure would be. However, this or like totally unfolded structures are out of distribution.
yeah.
Yeah.
I
Okay.
have
Yeah.
I have one small question about the training. So, during the training, what's the distribution of your data set? Do you contain a lot of similar data samples, or do you have varied number of data samples ranging from a lot of disordered regions, then some structures are folded, or oh, my major point is that what kind of distribution do you hold? Small proteins, medium proteins, large proteins, and how do you decide that it's not overfitting and you still are getting the results that are very generalized?
Yeah, a really good question. So we use the CAT, which is a data set of protein structures, representative structures, uh, clustered by their fold. So we divide our train and eval with the fold classification. So the evaluation is always on fold that the training has never seen before. So the cats normally have like a distribution of, I think the average length is around 200 something. Don't quote me on that, but around that range. And they are, I would say, more folded than not. So they are they're classed by folded. They're fold. So most of the CAD structures are more well folded, I would say. But
Mhm.
the data set is weighted by the CAD classification. I hope that answers the question.
Oh yeah, thank you.
Yeah. So this is to guarantee that the model has generalized, not just memorizing what folder has seen before. So these results are on the validation set. Um, so we also in training tested ways to quantize or discretize this residue latent. So we tried LVQ or the VQ vector quantization and found that it actually does not work very well with this training scheme. And what actually worked is just using post hoc KNN on the latent space once the model is trained. So you can just use the trained model to embed the entire cath and then cluster the latent based on that. And then we can evaluate this trained SLAE autoencoder as a protein structure tokenizer. So there's a recent benchmark on this topic called structure token bench. Essentially, you give a structure and pass through some protein structure tokenizer, which in our case is a SLAE. And then it basically adds a probing layer to train the model on some supervised tasks. So the goal is to compare all these protein structure tokenizers across the same set of tasks in the same training setting. So here I'm just showing the best baselines, which are full-seq ESM3 structure autoencoder and amino acid, which is the one struct token bench proposed. So I'll say that the SLAE autoencoder latent having sidechain atomic information outperforms in most cases the baselines. So the baselines include two types of tasks that we focus on. So structural and functional site prediction. These are binding sites, catalytic sites, conserved sites, epitopes, and repeat motifs. And then there are a more biophysical task, which is the residue level flexibility prediction. So that predicts the RMSF, B factor, and NEQ. So all of these are operating on one residue token at a time. So this gives a the confidence that SLAE is representing its local residue in a meaningful way that allow these tasks to be successful. Okay.
Can you do any, are there any like atom level tasks? Like, like what is like a lytic site, for instance, and like knowing that?
Yeah, oh, this benchmark did not include atomic tasks, unfortunately. However, we do include a task that's atomic in this transfer learning scheme.
Hmm.
Yeah. So basically, transfer learning is a more broad kind of aspect of what I just talked about. Essentially, we freeze the pre-trained encoder. So latent would always be the same. And then the decoder transformer gets a trainable head. And that can be piped to task-specific data sets to be trained on or fine-tuned on. So we use the same set of weights for all of these tasks and just fine-tune on a specific data set. So the point is that the SLAE learned representation and this pre-trained weight actually transfer very well across all these different tasks. And I want to emphasize that they're on like different level of resolutions. So we have homology, which is fold classification, that measures the entire structure. So that's on an average pooled structure level embedding. And then we have the protein-protein binding affinity, which we let the head look at the interface, the residues on the interface. And then we have mutational stability, which we just look at the residue embedding of the mutated residue. So on the left, the scheme of comparing two embeddings from two different structures, and you can do a Siamese or contrastive way of learning from them. And then finally, we do have an atom level task, which is the chemical shift. So this is the NMR prediction task that would predict the chemical shift of the backbone nitrogen atom. So I think this is a really good example because NMR chemical shift, especially the nitrogen, is very hard. It takes a lot of impact from like nearby atoms, especially like ring current and other more complicated physical chemical factors, and that is what makes this task interesting. And we're interested to see if the SLAE can work on that. So these are the results. So overall, we're seeing competitive performance or even better to the state of art. So we're comparing the SLAE fine-tuned version to all of these best models that we can find that's published. So for example, this chemical shift, we're getting a lot lower RMSE and then higher correlation compared to all the other tried and true method that people have used before. So this, I think, is a really good indicator of the generalizability and how useful the SLAE latent trained only on first principle atom and distance can be. And I want to also suggest that we train two different types of head. Like you can do it for example, the affinity. If you do it without the interface and just look at the entire structure, performance is actually not that great. However, if you let it focus on this interface, the performance increases. Same as the thermal stability. So if you do not focus on the mutated side, the performance is okay. But if you have an emphasis on the mutated site, that's what gives you the performance boost over the previous state-of-the-art method.
And what do you mean by like focus? Like what do you do? Is it like you're just using those residues or are you like
So you extract the latent feature and feed it directly into the head.
okay, so you're just using the features of that of the like of those at the interface or something.
Yeah.
I see.
Okay. All right. So we tried these tasks and it was really great. And that kind of answers our question of can we get a representation that's good for these predictive tasks? And I want to just zoom out and just interpret the SLAE latent space a little bit and try to figure out why it's helpful for these tasks and what future work can we do based on the how this latent space is organized. So we basically just took the cat embedding, the residue latent, okay, and just projected onto the UMAP. And we're looking at the organization of this latent space in interesting ways. On the left, you can see that it is organized by residue types. So it is kind of expected that these clusters will have a dominant residue type and residues with similar chemical properties will be clustering together. So for example, hydrophobic are here, and then we have these specials were like all around. And on the right are more interesting ones. So it's also kind of clustered by secondary structure, and that has something to do with hydrogen bond. And it's also organized along axis of solvent exposure. So we're seeing that this latent space has a lot of desirable organizations, meaning that it is performing what we wanted it to do.
I wonder how it's not trained for this task, but like if you think of Blaine, because you are basing it off of these like, local environments, and I wonder how that performs in terms of like retrieval. So if you think of like fold seek or any of the like later versions of like you're trying to learn some tokenizer over a 3D structure, and your actual task is to find other similar sort of proteins or protein families. Um, how what you learn is like how sensitive is it, and like if it can retrieve sort of things that are relevant.
Yeah, that's a really good question. We've been thinking about it. And I think one use case for it is actually motif retrieval. So searching for
Hmm.
similar motifs that we're interested in. And I think from the limited test we did, it works. But we haven't done anything like systematic that
And
evaluate.
is 8 angstrom enough, or do you like is a motif sort of defined on a bigger vol like a radius?
So 8 angstrom I think is enough. Especially if you're looking for multiple residues in one motif.
Hmm. Because like it seemed like you had issues going bigger than 8, right? When you're
Yeah.
training.
Yeah, computationally, yes.
Hmm.
And that's
Interesting.
kind of unfortunate. We did try 10. The performance was okay, but the model was taking a long time to converge, so we can't say whether or not it actually improves or not.
I see.
Yeah.
Cool.
I have a follow-up question on
Yeah.
that 8 angstrom. So I understand that you take a fixed radius and then select the neighborhood atoms. But what if in some protein the neighborhood atoms exceed? So you need to fix the length of the input, right?
Actually, no. So we're not fixing the number of input. It's a radius cutoff.
Okay. So
So
if
the average number is around like it can range from like 100 to up to 300 like atoms in one graph, but we do not remove atoms. So all the atoms in the neighborhood is accounted for.
so, do you use a different um, length encoder every time, or how does that happen?
So it goes back to this. Right? So it happens every time with this parallel kind of architecture because you're looking at these graphs all at the same time. And what you're only doing is essentially sum products and tensor products. So you're actually not working with like tokens at this stage. It's a graph. So you have a fixed size graph to start with. And if you want to batch it, yeah, it comes in different sizes because protein are have different numbers of atoms. Yeah.
I mean, obviously this is a future works kind of thing, but I think a lot of the gains you would see on things when you have now small molecules, interacting with the residues, right?
It's right in my future works. But yeah,
Yeah,
yeah.
yeah,
We're very interested in that and actively working on that aspect.
yeah.
Okay, go back. Another thing that sort of inspires our future work is we see that the latent has a way of discriminating native versus decoy atomic environment. So um, we take a data set of 130 native structures, and that was augmented with thousands of decoys. And we basically project that all onto this SLAE latent space. So on the left is the protein level embedding space, that's the average over all residues. And the yellow ones are true native. The red ones are closer, better decoys, and the blue ones are bad. So this is just sort of showing that the latent distance on the PC, on the first two PCs, kind of correlates to the coordinates distance. And then on the right, I think what's more interesting is that this also applies on the residue level, which was not explored before. So on the residue level, we also see this sort of organization, that the ones that are closer to the real ones are bunched together on this latent space. And then we can just simply train like a logistic regression model on these residues to let the model decide, oh, is this a native or is this a decoy? So we're actually getting some signal from there. So I think this is some sort of indication that we can have scoring models developed on the per residue atomic environment representation.
But what's the point of discriminating between native versus not?
The point is, sometimes we design structures, for example, and we want to
Yeah,
see if a structure is good or bad.
I see. And so
The
you
idea,
mean if it fools the model as looking native, you would say it's a good design.
that's right.
Okay.
And the hypothesis is, again, compositional. So if all the residues are happy, the structure itself should be happy. So we're
Hmm.
sort of decomposing the problem into these per residue problem. And it also is a way to tell us, like, something's wrong with this residue. We should look more into that.
Yeah, I mean, I
Like,
wonder what how it would do as like a filtering method in general. Like if you, because right now, let's say you take the design pipeline where you have some structure, you redesign it, you fold it back, right? Let's say you use folding as a like a filter, as a consistent or not of your design, and then you test whatever. So if you take those that have been, at least that people have tried, and you know, like, okay, well, these de novo proteins worked versus these didn't. And would you be able to sort of predict that from this?
yeah, we have a lot of these data-based prediction tasks going on right
Okay,
now. So using these SLAE latent. Yeah. I think that what I can say is that there is signal that we can train models based on that. Yeah. And it's actually the very reason why I designed this latent space. I want to have better filters than just AlphaFold. So,
sure.
yeah. And then this is another aspect, like how we can use this to help our design pipeline. So on the left, I'm showing the FID metric for image quality assessment. So you can decide how far in terms of distribution your generated image is from a good set of true images. And then Tianyu from our lab proposed the protein distance for FPD in this case, in that you can measure the distance of, for example, the native cat to a sampled ensemble of from your allatom protein design model, for example. And that tells you how far your model has drifted away from your training data set. So on the right is, so previously we can use some sort of either sequence embedding or backbone embedding model to sort of replace the inception model in the FID. However, with this all atom residue level embedding model, we can look in this even further. So you can now split these distributions by, say, amino acid type. Here I'm showing 3I-LNN, and then compare that to, say, all the I residues in the cath. And how does that look? How far the distance is. So it's essentially comparing, is my all atom generative model recapturing the same atomic environment distribution in my training dataset. So it's able to reveal some of the interesting things that these generative model has learned and biases.
Hmm.
And then finally, I want to show one of the cool experiments that we did on the SLAE latent, which is latent interpolation. And then recall this autoencoder representation, right? You have a manifold that you project your residues to. In this case, for example, I have the ADK protein. That I can start with two conformations that we know, state A and state B, and then interpolate between their latent linearly on the latent space for each residue. So essentially, it's a linear interpolation for the two proteins on this latent. And we can match that to MD simulation frames pretty good. And then I'm showing this little video of the linear interpolation. That includes all the side chains, which appear to be reasonable. So we can say that linear interpolation between residues decodes to valid intermediate structures for the cases that we tested without explicit smoothness regularization. So this means that a lot of smooth latent came in, for example, in VAE came from some KL regularization or other methods. And we designed
Yeah.
this latent to be smooth with all these laws and input design and hope that it could be something that's smooth across these atomic environment and it kind of behaves like what we expected.
Do you see like the that matching with the energy that you predict, or the energy that you predict matching the some like, computed energy?
So we can only predict pairwise Rosetta scores, which is not the energy of the structure itself. So unfortunately, there is no matching to MD energy, for example.
Hmm.
But that's being worked on. So we are working on energy functions on the latent. But also, I want to note that this is a more rigid body movement. For cases that involves unfolding, we're seeing some pathologies in the unfolded state, which is kind of expected because the training has never seen unfolded structures and the decoder struggles.
Sure.
But I also would compare this to local backbone autoencoders, for example, the one used in ESM3. So we do the same experiment. This is a CalModulin, and we interpolate, and the SLAE looks good, it's smooth, like played in the video on the right. And the ESM3 have these pathologies, like the conformations jump in steps are right next to each other. So it's kind of snapping back to its training data set a little bit and have these jack transitions. And and we think that SLAE produces a smoother and more physically consistent interpolations, where and we can also model sidechains explicitly. So with all these things that we learned about this latent and the behaviors, we're pursuing these future directions. Like like you said, the ligand and small molecule, and we're interested in binding and active site modeling. And we're developing latent scoring functions to distinguish good and bad atomic environment. And finally, we have the latent modeling track, where this design of autoencoder lend itself to these latent generative models like latent diffusion, autoregressive, or some sort of hybrid. And down here, I'm showing some early results from a small autoregressive model, like GPT style, trained over the discretized codebook that we got in training. And it's starting to generate some reasonable refoldable structures. So that's pretty interesting to see. Yeah. And just in conclusion, the motivation of SLAE is that we want an all-atom protein representation for downstream prediction, quality assessment, and generative task. And we approach this by having the strictly local atomic environment encoder coupled with global structure decoder in this hard way supervised with energy regression for a rich and generalizable residue latent space. And to summarize, it learns a compact all-atom protein representation as we have hoped, and balances local chemical detail with global structure coherence with our design, and its latent space is interpretable and transferable. And by that, I would like to thank my collaborators, Tianyu, which was really helpful, my PI, Posu, Jojo Zhang from University of Wisconsin-Madison, and Hannah, that's his PI that worked on the chemical ship prediction part, and all my other lab mates in the postal lab. And I think that's it, and I'm open to further questions.
Awesome. Thank you. Great talk. Alec, I guess, has an additional question. So are you using autoencoder, or like I guess a vanilla autoencoder or a VAE? And
Yeah,
why?
so that is kind of I talked a little bit about here. I'm using a vanilla autoencoder. So we tried some version of VAE and actually didn't work that great. And instead, we opt for smoothing functions using the laws like auxiliary task. And it turns out that our latent is smooth enough and it kind of just self-regularized. Yeah.
And then some more testing time questions. What's the inference time? Um, and then more generally, will the code model be available?
Yeah. So the inference time is very fast. If you go through the entire encoder decoder, the bottleneck is actually the encoder, which is a graph. But I can tell you it's not that slow. So rest assured. And yeah, we're getting the code to be publicly available. But also in the meantime, feel free to reach out to me if you need early access or talk about collaborations. I'm definitely open to that. But the code will be open sourced.
And when you say fast, like what's the scale of?
Let's say if we were to embed and then decode out 1,000 structures, that takes about 5 minutes on an A100.
Okay, cool.
And you can
Um,
parallelize that.
yeah. Hmm. It's okay. So a bunch of questions remain. What part is equivariant versus sequence dependent? He says all atom must be equivariant, but I imagine residue level latent is a sequence ordered vector. Yeah. How do you enforce that at train time?
Uh, I kind of want to see the question again.
Yeah,
So
yeah.
the question was that the encoder is equivariant. Is that?
Yeah, because sorry to
Oh,
explain, it looks like you're using the residue level
right.
right latency, and so I imagine this is sequence dependent. How do you enforce that given that the structure can be ordered differently?
Yeah. So in the decoding time, you actually give it residue numbers. So it's
Okay,
just a
got
rope.
it.
Right?
Okay.
Yeah.
So the atoms can be ordered anyway
Yeah.
because it's a
Yeah,
geometry,
they
but
can be
then you have
ordered
to
anyways. Yeah.
enforce. Okay.
Yeah.
And does it mean you have so for each atom, you know which residue it's which amino acid it's part of, and you give it a number that's sequential.
Not in the input.
That is the input on the decoder. Okay.
So the
Only the
decoder
default.
tries to guess like which atom is available, for example,
Okay. Got
and
it.
also where it is.
Okay. But then once you use the encoder for tasks that require the sequence, you pass the sequence along.
So for tasks such as these downstream tasks that you're saying, sorry, let me go back.
Yeah, maybe maybe I misunderstood, but it looks like you're using right the latent for each of the residues here, right?
Yeah, so you have a frozen encoder that project the residues into these latent, and it's the same decoder that we train for decoding the structure. You're just tagging on a different head.
Okay.
So instead of decode out sequence atom coordinates, you're getting affinity predictions.
And here you mean pulling the each atom's embedding into residue embeddings.
Yeah, after the encoder, before the decoder.
Okay.
And then after the middle layer, which is latent, you actually project it up back to the decoder's size. So
Okay.
the transformer has to be wide to reason.
Got it. Thank you.
Really great questions. Thank you for asking. Yeah.
So then question about training, co-train the global structure model with SLAE training, or is it one-way information flow? The
I
global,
think um let's see. So in this flow, it is one way in terms of forward propagation. So you give it the atoms and distance, and you decode out the structure and the energy, and the back propagation is through this reconstruction and energy losses. I don't know if I'm understanding this question correctly, but
yeah, I'm
yeah,
not sure. I mean, I think like the SLAE training includes structure, the
yeah,
global structure prediction.
yeah. Yeah, that's what you essentially supervise on.
Yeah,
That's mainly a self-supervised method of learning, though you are weakly supervised by these energy terms, which are properties of the input structure.
yeah.
Okay, so
Uh,
so just I just missed. So you don't use results from evolutionary sequence models or something like that
Yeah.
as an input.
Yeah, so this is the key point is that it does not depend on any evolutionary information. So all you have is structure and you extract atomic information that's helpful for a downstream task. So it has never seen MSA, for example, and it does not know things about evolution.
So like models like AlphaFold or Boltz, you're not using the
No.
data as input data to train a model.
So we train on synthetic structures predicted by folding models, but I don't think that would give the SLAE some sort of evolutionary knowledge. The training input is purely structure.
There's
Uh,
no MSA or anything like this, right? So
yeah.
Yeah, but you, because of distance cutoff, you don't account for like if you fault long distance interaction or potential long distance interaction.
Right. So the encoder has no knowledge of long distance interaction.
And can you somehow produce this or infer or predict long distance interaction? I just not in the scope of this project.
Right now it's not, but I think the assumption is that if we can get the global structure correct, it has learned some sort of long range ha- long range relationships between these residue tokens, if that makes sense, in terms of structure.
Right. So you already assume some folding configuration,
Yeah.
right?
Yeah. So
Okay.
it has to learn some sort of prior for fold.
Okay. Thanks.
Yeah.
an additional question is, would it be applied to secondary structure prediction? Presumably.
Yeah, I think that's reasonable. But though it's like you give the model input of the structure. So you
Yeah.
can easily classify secondary structure that we've tried. And then you can also see that they are already organized by secondary structure. So it's actually pretty easy task for the latent.
I like also asks about like active learning from some experimental data.
Yeah, that is a very important part of the downstream work. We're trying to do the lab in a loop with experimental data and try to augment this pipeline to
And
eventually get,
that's for like for the filterings of designs. Okay.
yeah, filtering of designs, say binding. And we're just trying to use this representation to inform us and then learn from the experiments.
Are you comparing against other methods or just to
Uh,
see if actually it helps?
in terms of experimental data, we're using our
Yeah.
own data right now is mainly for our projects that goes into experimental testing. But
Yeah.
I might I might think about like comparing with other methods if there's a reasonable benchline, yeah, a benchmark that we can work on.
Yeah. Because it'd be still interesting to see if it actually translates into better performance or
Yeah, so I think that requires a more generalizable scoring function, which is something that we hope we can get from a larger, say, native decoy data set. If we
sure.
have a larger scale, then we can try to train a more generalizable version of that.
Cool. Very cool. Any additional questions from anyone in the audience? All right. Well, if not, then thank you, Yilin, so much for the talk.
Yeah.
Um, and have a good rest of your days, everyone.
Yeah, thank you for coming to the talk. Thank you, Peter, for reading all these questions. I couldn't see them.
Oh, no
Presenting.
worries.